Is this working?
That's looking good.
Perfect. So as Graham said, my name is Sara Gago and I'm actually a lecturer in fungal disease biology in the Manchester Fungal Infection Group at the University of Manchester. So today I'm going to talk to you about how the interaction between different pathogens shape the outcome of respiratory diseases, right? Because as Graham says, most of us, when we get an infection, we are not always getting a single infection. Sometimes we are treated for multiple infections and we don't know who's coming first, if the order is important. And the problem of these interactions is that sometimes when you are giving an antimicrobial to treat one of these infections, that produce like a lot of interactions with another drugs that you are given to treat some other infections, right? So there's a problem there, and there's a clear need for understanding how these polymicrobial infections, that is the way we call them, work together. So everybody who is here in this audience today, all of us have suffered from respiratory infectious diseases at some point in our lives. There's no one who can escape from this, and indeed children, right, start with this viral bronchiolitis just basically when they are just born, right? So most of them are caused by bacteria. You might have heard about bacteria pneumonia, viruses, we all get flu. Most of us will have had COVID during the last 5 years, at least a couple of times, right? And fungi that, as you all know, Aspergillus fumigatus is the major driver of lung fungal infections. But as I mentioned, in the last years, and in particular in the last 5 years, it has become evident that a significant number of these infections are polymicrobial. What do we mean with polymicrobial infections? We mean that they are caused by a combination of different pathogens. These combinations can be to different bacteria, to different viruses, to different fungi, or it can be a combination of all of them, right? You can get a bacteria pneumonia and a fungal pneumonia. You can get virus with fungi. And what that does is an absolutely mess in our lungs. Most of we know about these polymicrobial infections came from the field and from studies in patients with cystic fibrosis, right? Cystic fibrosis is a chronic lung condition where some patients have mutations in a gene that makes them more susceptible to recurrent infections because their lungs defense don't work properly. So basically in these patients, right, you get a diverse colonization of microbes, mostly in the lungs, and the composition of those microbes that are in the lung can change because of the antibiotics that you are normally treating, and that can lead to the overgrowth of some microbes that are more pathogenic. And with that overgrowth of pathogens that are more pathogenic, you can get worse disease symptoms, you might need hospitalization, and most of this is because there's a competition of the different microbes that are colonizing our lungs. In the context of aspergillosis, I made here like a diagram. These are the most common polymicrobial interactions or infections that we know in aspergillosis, right? We have what we called influenza-associated pulmonary aspergillosis, that mostly occurs in immunocompromised patients, but also in some immunocompetent with unknown risk factors. We have what we called during the COVID pandemic coronavirus-associated pulmonary aspergillosis, which is quite rare and only has been described in patients who are in the ICU with COVID-19. In cohorts of patients with lung transplant, it has been described RSV-associated pulmonary aspergillosis. Quite strange, right? But it happens many, many months after the lung transplant in many situations. So it's something that we need to be vigilant. So then we have combinations with bacteria and fungi, right? We have Pseudomonas and Aspergillus in cystic fibrosis. I have a question mark here because the evidence is a little bit contradictory, and I will show you later on what we know about these interactions. And then we have in patients with chronic pulmonary aspergillosis, you know that one of the most common risk factors is a prior history of tuberculosis or NTM infection. And we also have, and according to the guidelines that has just been published in Thorax last month, that common seasonal viruses, what we mean with common seasonal viruses, those for which we all get vaccines, right? Flu, RSV, the coronaviruses that are not COVID-19. And so those can exacerbate allergic bronchopulmonary aspergillosis, but we don't know when those flares happen, we cannot predict them, and we are trying to work a little bit on that. So for the purpose of this talk, I'm just going to focus in 3 case scenarios that are the ones that I've been working in the past, that it's influenza-associated pulmonary aspergillosis, cystic fibrosis, and chronic pulmonary aspergillosis. So as I mentioned to you, invasive aspergillosis is typically a disease for patients who are severely immunocompromised. That means patients who are on chemo because they are having like a cancer process, patients who are on long-term corticosteroids, etc. So in 2018, it was described for the first time that the diagnosis of invasive aspergillosis can occur in patients with severe influenza that do not have any underlying condition. That was a revolution in the field, right? Because we always said that patients with aspergillosis either had a chronic respiratory condition or were immunosuppressed. But when we go back to the literature, the first time a co-infection between flu and aspergillus has ever been described, it was in 1952. So it was not something that we could say it was new. As I mentioned, this study published in the Lancet Respiratory Medicine in 2018 has changed all what we know about how aspergillus infects. And this was a study carried out in Belgium and in the Netherlands when they include patients from 7 ICUs between 2009 and 2016. And what they found, it was that in those patients with flu, 19% of those develop pulmonary aspergillosis. Importantly, 32% of those were immunocompromised, so they have the risk factors that we knew that might make them more susceptible to aspergillosis, but 14 of them didn't have any underlying condition. So what we've done so far, and people have been doing so far, is trying to explore whether treating patients with flu with antifungals before ICU admission might decrease the incidence of invasive aspergillosis in these patients. And you will say, Sarah, but if you get a flu, why are you going to get aspergillosis, right? Why is that happening? So there are a few studies that suggest that 3 main mechanisms might be occurring, right? The first one is that the virus, when the virus infects the airways, they replicate in a type of cells that are called epithelial cells that cover all the lung surface. So it's thought that the replication of the virus in that cell, it's gonna kill the host cell, and that makes aspergillus easier to germinate and penetrate into the lung tissue. It has also been hypothesized that flu can affect some immune responses, and in particular, it can make macrophages, that are cells that when they see an Aspergillosis spore, they capture it and ingest it and kill it intracellularly, right? So it's thought that flu can impair that mechanism by which our immune system can kill the fungal spores. And the other mechanism that has been proposed is that the virus can affect some of the clearance mechanisms in our airways, so Aspergillus can persist and colonize and potentially germinate. And you would say, okay, Sarah, you said that patients with flu have aspergillosis and patients with COVID have aspergillosis, but are the mechanisms exactly the same? How frequent is that? So COVID and aspergillosis is far less prevalent than aspergillus and COVID. And indeed, we did a study during the first COVID wave here in Manchester, and we were only able to find one patient who had this co-infection during the first wave. And no patients have been reported ever since in Manchester. So the thing is that even though the progression of the disease seems similar, it's thought that in patients with COVID, the risk of developing aspergillosis is associated with the treatment with corticosteroids, because at that point of the pandemic, when all these cases started to be reported, all patients were getting high doses of corticosteroids to suppress the inflammation caused by COVID. However, as I mentioned, since the COVID slowed down, I would say, I was about to say ended, but nothing has ended, slowed down, the number of cases reporting COVID and aspergillosis has dropped significantly, and it mostly occurs in patients who have a very serious condition in the back. So the other example I wanted to mention is those interactions between Aspergillus and Pseudomonas that I told you that occur in patients with cystic fibrosis. And some patients with ABPA, they either have asthma or cystic fibrosis, an underlying condition. So we know that both pathogens can affect lung function, and it's suggested that their combined presence can have even an even worse potential. However, as I mentioned, some studies are contradictory, and it depends on the way these research studies have been set up. So if we grow the two microorganisms, one and the other, in culture media in the lab, it seems that Aspergillus might be secreting some factors that inhibits the growth of Pseudomonas. But this completely change when we, in our model systems, we incorporate host cells, right? If we have immune cells in the system and we do this co-exposure experiment, we get that Pseudomonas grows better in the presence of Aspergillus. If we move into in vivo experiments and clinical studies, we see that patients with cystic fibrosis, where the two microorganisms are isolated, you get a synergistic effect. That means that the presence of the two pathogens makes them grow quicker. So that makes that your lung function can be reduced and your prognosis is also very poor. What is thought to mediate these interactions is what I said, that it seems that Pseudomonas and Aspergillus are secreting factors that might help the other counterpart to grow better. It has also been described that the two pathogens can create like a film on top of your lung surface, and that makes your antibiotics and antifungals very difficult to penetrate in the tissue and to clear them out. And it has also been reported that co-infection can amplify the host response and tissue damage because you get a lot of inflammation. So it's like in COVID when they were talking about this cytokine storm, right? That they were saying on the news all the time that patients were getting a lot of inflammation because your cells are very active to try to fight the infection. So since I started here my first postdoc until I moved to my lecturer post, I've been working a lot in patients with chronic pulmonary aspergillosis. And one of the main focus in my lab at the moment is to try to understand why and how a prior history of a mycobacteria infection or a concomitant infection with a mycobacteria can affect the outcome and the progression of chronic pulmonary aspergillosis. What we know from the literature, right, is that mycobacterium and mycobacterium tuberculosis and non-tuberculous mycobacteria can mimic or overlap with chronic pulmonary aspergillosis because the symptoms radiologically are very, very similar, and which makes the diagnosis very complex. It has been thought that aspergillus can grow in the cavities that tuberculosis lives in your lung, and that might create an environment for the fungus to grow. But not all patients with TB have cavities, and not all patients with TB and chronic pulmonary aspergillosis have cavities on their lungs, so something else must be going on. So what is most important in this context is that this combined infection is not uncommon, and there are a lot of challenges therapeutically, in particular because rifampicin, that it's the antibiotic that you use to treat tuberculosis, cannot be prescribed in combination with itraconazole because of drug-drug interaction. So in my lab, we are trying to understand how the immune response against those pathogens in separate and in combination work to try to develop new diagnostics and treatments. As I mentioned to you, that what we thought that it was that the structural damage is a shared risk factor for the development of long-term tuberculosis and also aspergillosis because it produces cavitation, fibrosis, or bronchiectasis. It's also thought that macrophages that have been infected with TB because the mycobacteria persist inside these cells are unable to pick up and kill aspergillus spores. So it might be the case that by that means the fungus is more keen to grow in the lungs. So what we are doing in my lab to try to address these questions is we are trying to set up infection models where we grow immune cells and we expose them to mycobacteria, and aspergillus is in different orders. So what we try to mimic with that scenario is that who is coming first, right? Is the order of infection important for the development of disease? And what happened to a macrophage when it's first exposed to a bacteria and then to a fungus? Is able to cope with the two of them? Is killing one more efficiently than the other? So we are also carrying out what we call patient immunophenotyping. That means that we are taking your blood and we are staining for different components that we know that are important for the immune response against these pathogens. So what we are trying to see with this is whether there is a component of the immune system that is not working properly in the case of co-infection. And we are also trying to look at your blood for some genetic markers to see whether any processes that we know that are linked with antifungal immunity, that is how the body fights against aspergillus, or antibacterial immunity, are impaired in these patients with co-infection. So to wrap up, and 20 minutes on time, Graham, this is never going to happen again. So what I want you to see is that co-infections with fungi and other microorganisms are very challenging to treat because sometimes the infections mimic each other, right? So there's not clinical suspicion on what's going on. And by the time we want to treat some patients, it's too late. That happened particularly in the case of flu and aspergillosis during the pandemic in 2018. The diagnosis center, there is a strong diagnosis and therapeutic challenging because infection complicates the diagnosis, and also you require like a very complex treatment strategy, and you need to know for how long, what antibiotic is better, and avoid these drug-drug interactions. And the final thing is that we think that what happens is that when you are infected with a pathogen, right, you are creating an environment that might facilitate the persistence of the second pathogen. And that is all I have to say to you today. And thank you very much for listening.
Thanks, Sarah. That was really interesting. Has anyone got any particular questions?
I have, Graham.
Tom, I think you were first.
Okay. Hi, everybody. Hi, Sarah. Thanks for that.
Tom.
Oh, sorry.
You mentioned
Tom, what did you have to say? Ask.
about Pseudomonas in terms of cystic fibrosis only, but I don't have cystic fibrosis, but I've had Pseudomonas in conjunction with the Aspergillus, and it's been very difficult to treat.
Yeah, it's very common. Yeah. So I mentioned cystic fibrosis because almost all patients with cystic fibrosis get like chronic pneumonia because of Pseudomonas. But of course, when talking with Chris Cosmidis, he told me that many of you also have very persistent Pseudomonas infections growing in your lungs, and it was extremely challenging to get rid of that. Yeah.
Yeah. No, I just wanted to check that so that you'd know that yeah, it certainly seems to be very common.
Yeah.
Yeah, and very difficult to. I was treated for it through a PICC line, staying at home and on a PICC line, but I had a severe reaction after 2 or 3 days.
Oh.
I had a severe reaction to the drug, and I spent 11 nights in hospital. And once they took me off the drug, the symptoms resolved. Like I had a severe fever and dreadful headaches. So the Pseudomonas was, I gather, it was kind of killed off for a while, but I think it's coming back again now, to be honest.
Yeah.
But of course, you don't really know until the sputum is analyzed, isn't that correct?
Yeah, exactly. I think that the way they do the diagnosis of your co-infection is just with a culture in sputum at the moment. I don't know if they are using something different, but I think that it's the standard that they use at the moment at the hospital.
Yeah, it's a slow process because
it's a very slow process, yeah. And also because what I find with these pathogens, before working at uni all the time, I was working in a clinical lab, is that even though if you don't get it growing in your culture again, right, you can never be sure that it's completely gone, right? So that is why it's very important that when you have signs or you are feeling a little bit under the weather, you contact your consultant to discuss this. Yeah.
Okay, thank you.
Evelyn, I think you're next.
All right. I was getting a similar question, question along the lines that Tom has just mentioned. My question is does it make it harder when you colonize? I mean, I've had I was first diagnosed with and treated for Pseudomonas in 1995, and within a few months, and not just an ordinary everyday sputum culture showed up the Aspergillus. So, but that was way before I became to be a patient at Winshaw. But then when I had successful treatment with the itraconazole for the Aspergillus around about 2010 or there, 2012, very quickly I developed a scedosporium, and I just, because the Pseudomonas is managed with nebulized colomycin, and I've only had one occasion when I've needed IVs for the Pseudomonas since then in all those years, which is good. But the Aspergillus and the scedosporium, it's like they play hide and seek. And I find it really frustrating at times, because it's as though it's not there, but I really feel as though there's something more going on than just the chest, the bacterial infections. So can the fungi hide and be difficult to find in cultures when you've got colonizing bacteria as well? Because I am colonized with Proteus as well. And that's a really big problem for me now.
Yeah, that's a very, very interesting question that comes again from how we do the diagnosis of these infections, right? So I'm going to try to explain it as simple as possible. So if I don't explain myself right, please stop it anytime, right? So when you get your sputum sample, right, you normally split it among, for this, so it's in different samples, and you can say, this tiny bit goes to the bacteriology lab, and they do the growth to look for your specific bacteria. And this tiny bit of the sputum goes to the mycology lab. And we used media that is specialized to culture that microorganism. What can happen is that, and this is one of my main frustrations all the years I've been working in the clinical lab, is that the sputum sample is a highly heterogeneous sample. That means that's all, when we split it, it's not the same all the time, right? So it might be the case that the aliquot that we are getting in the mycology lab doesn't have all the microorganisms that we want to look for.
Yeah.
So for that reason, your results are not very consistent over time. And on the other hand, it can happen what you said, right? So microorganisms that are more adapted to grow in culture and grow quicker can hide other microorganisms that are not as well as adapted to grow in the culture. So what you said, it's like it seems that they are playing, but sometimes it's because of how the cultures are set up. And that is why the trend at the moment is to try to develop molecular tools that might be able to help and improve the performance of the culture. But at the moment, culture is the best option we have, unfortunately. Yeah.
Yeah. Oh, thank you. Thank you for that.
Could I just butt in quickly? So, do you know, that's just made me think. Do you have any feeling for whether biofilms in the lungs play a role in that? So, like if you're coughing up sputum that's getting tested, do you know whether that's kind of the same mixture?
Beth is frozen for me. Is it working for you?
No.
Yeah, we're having a few issues, I think. Is it everybody? I thought it might just be me.
No.
No,
We've lost,
you're fine.
I've lost Beth.
I lost her. Yeah.
Yeah.
I think she's probably dropped off. Janet, do you want to ask your question?
Well, yeah, sort of. Mine was similar to Tom and Evelyn. So I was diagnosed with the Pseudomonas, and I've also got asthma and Aspergillus. But so is the culture the only way to determine the different conditions or path that you might have?
So I think at the moment, because this depends hugely across different countries, right? So I did my training in Spain and we were using culture very little. We were doing all like molecular assays, but I know that at Withenshaw in the mycology lab and in the UK in general, the trend is to use culture and just to do PCR for a limited number of cases. So I'm quite sure that in the mycology lab at Withenshaw, they do Aspergillus specific PCR, that is a molecular test in the sputum. I cannot comment what they do in the general microbiology lab because I'm not very familiar to how they do it here, unfortunately.
Yeah, I was just, this was just very recently, and I took was it ciprofloxacin? Is that how you say it? Yeah, so
Yes.
I do feel a lot better, so I'm gonna talk to my consultant next.
Ciprofloxacin, I think. Yeah.
Yeah.
Thank you. Yes, thank you, Graham. Next week, so I guess I have to do another sputum sample, and we'll see what happens.
Yeah, possible. Yeah.
Yeah, thank you.
Can I just come back in there
Yeah.
again, Sarah?
Sure.
Go ahead, Tom.
In terms of Pseudomonas, and I know about getting it tested and so on, but when you look at sputum, is there any way that you can sort of know pretty much straight away, yeah, that's Pseudomonas or that's whatever? Because when I had the Aspergillus, when it was at its strongest before I started on itraconazole and that, like it was very stringy mucus, like twine, right? All the time. And now the mucus is more, I don't know, lumpy and liquidy and green, and it's probably a hard question. I don't expect an answer really, but
so the thing is that it could be ideal, right? That you can look into your sputum sample and some sort of predict what's going on, but it's very sometimes from the color you can predict there's something going on, but you cannot tell what is going on until you get your microbiology results. And in particular, because many of you are treated also with mucolytics, right? To try to make to facilitate the expulsion of the mucus and things like that. So it makes the mucus is a particular nasty sample to work with, right?
Yeah.
okay.
Can I ask another question, Sarah?
Sure.
What is the definition? You know, when they say there's pus, I know what, purulent, but then sometimes you say there's pus present. So what defines that, please?
Oh, I don't know that. Graham, do you know that?
I think it's again, it's down to different clinicians expressing it in different ways, and for different reasons in different contexts. So purulent is obviously quite an advanced infection. I think traditionally it was one that was defined that it started to smell, that type of thing. But I don't know whether it's one of those words that's quite old-fashioned, and maybe it doesn't mean the same as it does today. But pus present is what it means on the tin. It's just there. I had a question for Sarah. If the internet will hold up long enough, I assume I know the answer to this question, but from what everyone's been talking about, we see these infections come and go, and that can be due to taking antibiotics. Maybe from what you've been saying, maybe if we get on top of the Aspergillus infection, we can reduce other infections at the same time. Maybe that's partly why people start to feel better. But we know Aspergillus evolves in the lungs as it infects, genetically evolves and phenotypically evolves. Is there any evidence that Pseudomonas and all these bacteria also evolve alongside Aspergillus?
Oof, I don't think I don't think I've seen any study where they do co-evolution of pathogens. That would be something really, really interesting, right? In particular, to see whether when you have the two things together, you get, I don't know, like a high mutation rate in genes that are important for your antimicrobial treatment. But I haven't seen any work in that sense for fungus and bacteria. I think there are some studies where they do some bacteria coevolution, but not with fungi and bacteria. Yeah.
I mean, it's almost impossible to separate them in the lungs anyway to do experiments, if you like.
Yeah.
I mean, we know they evolve resistance to antifungals.
Yeah.
That's the thing we take notice of. But I'm just wondering whether you once you've treated, say, a pseudomonad once, and it returns 6-8 weeks later, is it the same thing that you're treating? I suppose we only really care if it changes into a resistant strain, but there must be lots of other strains, and who knows?
Yeah, there are some papers where they do evolution of the microbiome in patients who are chronically colonized, but yeah, it's just like a global view on a metagenome level. Yeah, possibly it's not the same thing.
Yeah. And yeah, interestingly, there was that news story 2 days ago where babies, I think it's the Lancet, so it's a very good quality study, babies that are exposed to bifidium, one of the BIF genes, genus of bacteria, experience 50% less fungal infection, or less lung infection, I should say, which is an extraordinary finding. I presume they're comparing it to babies, newborn babies, who are not purposefully given some form of exposure.
Yeah.
It just illustrates that what you're working on has very, very, very primary roots all the way back to the first hours of our life.
Yeah, there was a study published like in Cell that it's like one of the most important journals of molecular biology, right? Where they exposed like neonate mice to RSV to mimic, the early life bronchiolitis that some of our daughters and sons had ever at some point had in their childhood, right? And they see that, for example, those mice that were exposed to this RSV they call it early life exposure, are more susceptible to develop fungal allergic responses.
Yeah.
So it's something that they call nowadays imprinting, that it's like a way to say that our immune cells, our immune system, once it has been exposed to something, it might be more susceptible to an infection for a second pathogen. Yeah.
Just illustrates the war that goes on, the microbial war that goes on inside us in our microbiomes. Evelyn, did you have another question?
The only other thing I was going to ask was, sorry for all these questions. The only thing I wanted to ask was, I'm not productive of a morning, but I am very productive as the afternoon and evening wear on. So when I do a sample, I usually do two samples, 2 pots. I always do it in the evening and keep it in the fridge, because I wouldn't be able to produce anything of a morning. Nobody's ever said not to do that, and but is that still as effective as if it was a fresh sample?
Yeah, because it's very likely that they will do the same with your sputum. They will collect it and put it in the fridge until the day after.
Ah, right. Okay. Thank you. That's good to know.
You're letting out our secrets.
No, that was really good, helpful, Evelyn. Thank you for asking that. I've never thought to do that, but I'll do that.
All right.
Yeah, just when you do that, let the consultant know, or the nurse when you drop it, and say, it's from last night because I couldn't produce it this morning.
I always put the date and time on my labels. And make sure they don't cover it over.
But you know, we always do that. When the samples come late on the day, you just put them in the fridge until the day after.
Right.
Okay, well,
Thank you.
obviously very important work. Thank you very much for your enlightening talk, Sarah.
Thank you.
If there are no more questions, I think we can let Sarah go if you need to get away, but you're quite welcome to stay and watch my tremendously interesting talk on vaccines, which will be coming up at 2 o'clock.
I'm going to finish my marking that I'm already late
All right then.
this time of the year. Your talk will be more interesting.
So I shall go probably fairly fast through this, but a lot of it's stuff you don't need to remember. Just remember the basic bits. I have a talk about vaccines to do, but first, just a quick glance at this, which is respiratory viruses, because there is a slight change. If you can see, this is a graph of the number of tests being done for a number of positive tests that have been carried out for respiratory viruses. In the UK, this is a matter of public record. The graph is available on UK HSA website, and it just shows you the main viruses that are monitored. In the UK, rhinoviruses, the common cold is more or less constant. Influenza dropping back. RSV, which Sarah was just talking about, is minimal as well, which is cool. A little bit of an uptick in COVID, and I remember one of the popular newspapers talking about a new variant sweeping China. Of course, it might be sweeping China, but it probably won't be causing lots and lots of symptoms. It's just a new variant of the same virus, which is spreading but not doing much terribly different to the other viruses that have spread. So I don't think there's much to worry about there. And this is valid up to the end of May, so it's reasonably accurate. I could have showed you other figures, but they don't seem to update them very often, and we're only getting to the end of April in a lot of them. So this is probably the most up-to-date. Little bit of a bump up on COVID to keep an eye on, essentially. If you needed an encouragement to get a vaccination, that's probably it. So I asked for questions before this group on the Facebook group, just so I make sure what I talk about is a response to what people actually want to hear. And someone brought up the subject of vaccines in Facebook this week. And I thought that's a good subject to talk about. I quite often decide what I'm going to talk about, partly from what people are asked for, and partly it's something that I don't know enough about, and I can do a bit of research and educate myself on them. So some of this stuff is new to me. Vaccine research. First of all, is vaccine research a viable option is a vaccine a viable option for aspergillosis? If you've got CPA or ABPA, the infection is already there. Aren't viruses usually preventative? Because we're all aware of the COVID vaccines, which are given prior to infection and are designed to stop infection. Well, if you've got CPA and ABPA, you're already infected. So what difference is it likely to make? What potential benefits are they likely to bring if we manage to develop them? And given that vaccines are a type of immunotherapy, it's stimulating your immune system to behave differently in order to prevent infection. There are other types of immunotherapy that we all know about that I'll touch upon at the end. So, in fact, there are several vaccines in use that are used to treat diseases that are already causing an infection, and they're referred to as therapeutic vaccines. There are a few examples here of therapeutic vaccines that you'll all be aware of. The human papillomavirus, which is now vaccine, is being given to younger people to prevent cervical cancer. That's a therapeutic vaccine and is working very well at preventing cervical lesions caused by papillomavirus, human papillomavirus. Because I think it's having we're showing in excess of a 90% reduction of cervical cancer in that age group, which is fantastic. That looks like one form of cancer that we've almost eradicated, which is amazing, because in my life, that's happened in my lifetime, which is fantastic. I used to work next to a group in cancer research that studied this, so it's particularly interesting to me. Oops. Yeah, so all that happens is they stimulate immunity to clear the infected or precancerous cells. So they still work even though the infection is already there. Hepatitis B is another one that we know a lot about. Same sort of thing. The vaccine aims to reduce the viral load, just as we would aim to reduce the fungal load, and restores the body to be more in control of this infection. They actually use them alongside antiviral drugs, so drugs that actually interfere with how the virus works, and using the two together can be particularly effective. And here at Wythenshawe, that's partly what the infectious diseases team spends its time doing, is treating patients with hepatitis. Viral hepatitis, I should say. TB also, most of us, I mentioned it a few months ago, have had the BCG and have got the little scar on our arms. That's actually a preventative vaccine, but it's also therapeutic. Well, BCG is a preventative vaccine, but there are TB vaccines which are therapeutic. They reduce relapse rates of tuberculosis, which is great, and they shorten the time that you have to treat, which is another benefit, and even control the infection that just sits there colonizing the lung tissue. It offers improvements in that. So it's certainly well worth having a vaccine, even for a very similar disease to CPA in many ways. And it just provide good benefits. And we even see these in use in cancer. And almost every month now we're hearing of successful examples of vaccinations, or interventions, I should say, but these are therapeutic vaccinations for all kinds of cancers. There is an FDA-approved therapeutic vaccine for advanced prostate cancer, which train the patient's immune system to attack the tumor cells. They work very well. I know, for example, the vaccine used to treat melanoma works particularly well on more aggressive melanoma tumors, and that's an amazing, within my career in cancer, melanoma was a deadly disease, and now it's coming under huge amount of more control from the medical side. So these, rather like the biologics we talk about, these vaccines can carry great potential for being effective. What about fungal diseases? There are therapeutic vaccines for Aspergillus and Candida, because Candida is a more common disease than aspergillosis. They're under research. All the research so far, there's nothing happened in people. It's all happening in animal models, usually mice. That's an indication of how early this research still is. And we had a talk from an American scientist in 2012 on an aspergillosis vaccine, and it's a measure of how slowly all this has gone that we're still talking about vaccinations in animals rather than humans, especially considering we were all witness to the speed with which you can develop a vaccine for COVID if you really make it a massive priority. In animals, these vaccines can enhance fungal clearance and modify human responses. That's what they aim to do. And there are a series of vaccines that are currently in experimental status. CAX1, ASPEF3, which is a particular allergen of Aspergillus fumigatus, and other types of vaccines, largely aimed at invasive aspergillosis, but should also be effective against CPA. So in order to decide what sort of vaccine we need, we have to sort of assess or whether how effective it'll be. We have to sort of assess the disease. So looking at CPA, CPA, as we know, normally occurs in people who have something wrong with their lungs, something physically wrong, a structural lung disease, old TB scars, emphysema, fibrosis, sarcoidosis, and so on. They have impaired lung clearance, so the fungus, any fungus getting into the lungs isn't cleared very quickly, and that offers the fungus a chance to grow. And we're now detecting more and more there is a slight weakening in the in the immune response in a patient with CPA. So there is a slight difference in the immune system of someone with CPA. Not enough to cause a problem with any other type of infection. Just seems to be this one type of infection that is allowed through the defenses. And we talked a bit about this a month or two ago when we talked about the research we're doing that provides patients with a boost to the immune response, and that seems to be working to keep CPA at bay. Sorry, I've developed a tickly cough. So, well, not always, whoops. Back. So patients are not always immunocompromised in the traditional sense, like a transplant or a cancer patient might be. But CPA patients do have subtle immune defects, chronic inflammation due to the infection. They tend to be treated with lots of steroids and antifungals. And underlying CPA is quite often things like diabetes, aging, poor diet, and so on. So all of these things are things that give the fungus a helping hand to infect us, and they're all potential things that we can attack with a vaccine. Okay, we know this bit. How does Aspergillus exploit these weaknesses? Lands in damaged lung tissue, it grows slowly, it protects itself using biofilms or as a fungal ball. These are things we've talked about before. Possibly we haven't talked about this one. It has mechanisms to evade your immune system. Maybe I need to give a talk about that because it has particular tactics that give itself an advantage in your lungs tissue. It can live in low oxygen, it can live at high temperature, and so on. I'll do another talk on that another time. And the body of a CPA patient hasn't quite got the ability to fully clear the infection. Due to all the inflammation, you don't get a fully productive cough, and you start to get fatigued, and so on. So CPA is not an immune disorder. It's though it isn't an immune disorder itself, it is seems to be a localized immune dysfunction. Something in the microenvironment in the lungs is not quite working right, so that allows the fungus to grow. There can be some small immune defects. We're detecting those genetically as time goes on, and Sarah has played a part in doing that for an immune defect for ABPA patients. And underlying infections. Okay, I'll try and go a bit fast. We're going all right. Okay, to summarize, I think we can remember that. Lung damage, it's not fully immune deficient, but there is a slight immune deficiency. How will a vaccine help? Well, one made for CPA could help by training the immune system to better recognize and control Aspergillus fumigatus as soon as it enters the body. So We think that maybe there's some delay, and a vaccine could stimulate a more immediate, more aggressive response, which would be great. Unlike a typical preventative vaccine, for example, those made for measles, this is a therapeutic vaccine after infection has occurred. So we've talked about that before. Two of the reasons why this is an advantage is to support the immune response and to reduce dependence on those long-term antifungal drugs. And of course, steroids, which nobody likes, nobody wants. Vaccine goals would be to boost fungal clearance of all the goog that is going on in your lungs. And specifically for CPA, this would enhance immune cell activity, especially neutrophils and macrophages, to kill or contain Aspergillus. So we want them to work harder, maybe work a bit earlier. Another vaccine goal could be reduce the relapse rate and the persistence, helping body suppress fungal regrowth after antifungal treatment ends would be a great thing for a vaccine to be able to stimulate. Modulate the harmful inflammation. So a lot of the problems that you experience with your breathing are not directly to do with the fungus that's infecting them, but to the inflammation surrounding that fungus. And when your lungs are inflamed, they don't work as well as they should. An interesting one is if we could control infection using vaccines, then that will give us another tool by which to reduce the fungal load in your lungs. And therefore, we would limit the opportunity of that fungus to develop a resistance to antifungal drugs because we wouldn't have to use them so often, which would be a great benefit. And even when the lung architecture is already compromised, a vaccine may be able to support patients with that lung damage. Why is it needed? Well, fumigated is everywhere in the air. We're breathing it in all the time. People with damaged lungs are everywhere. Antifungals themselves are long-term, and it's not particularly comfortable or attractive to take antifungals long-term. It can be kind of its risks and the cost to quality of life as well. And antifungal drugs are still, although they've come down a lot, quite costly. So that a vaccine would eliminate the need, a good vaccine would eliminate a lot of that problem. Hopefully a vaccine would be long-lasting immune-based control. So ideally, you would hope to, like other vaccinations that we have, let's say a COVID type or a flu vaccine, we would give it maybe once a year, and that would do for the whole year, and stimulate your immune system to control your CPA, for example. We would probably, it's most likely that a recombinant protein type of vaccine would be used, so very similar to CPA, in which a little bit of the recognizable surface protein of a fungus would be present in the vaccine to stimulate this response. We might, even though if it's available, we might use the vaccine in combination with antifungals for the most effective therapy. We've done that. Unfortunately, that's all the theory and that's all the research. Unfortunately, there are currently no licensed vaccine available specifically for CPA or indeed any other form of aspergillosis. Research is actively ongoing, though, which is good, and they're at the animal stage. These efforts primarily focus on preventing or treating invasive aspergillosis, but the findings may eventually benefit CPA patients. In fact, the same, an invasive aspergillosis vaccine might have a different target because we want to stop a rapidly growing infection. But if something can stop a rapidly growing infection, there's every chance it will also stop or benefit a slow-growing infection such as CPA. Those vaccines that are currently in research stage are the so-called KEX1 vaccine, It's currently in mouse models. Vaccination leads to robust antibody responses, which is great, and significantly reduces lung fungal burden and mortality, which is fantastic. So again, we're talking about mortality, and that's really relating to the acute invasive forms of aspergillosis. This seems to be a terrific candidate and one that's gone forward to further research. It demonstrates the potential for protective immunity. It's certainly, vaccines do work in mouse models, which, as we know, something that works in the mouse model has got a good chance of working in people as well. So that's a really good candidate, and I would hope that that would start to be tested in humans fairly soon, although we'll talk about that a bit later. The ASPEF3 and ASPEF9 antigens are a constituent part of another vaccine, VCVax, which is in development. Again, I think we're talking about mice. Again, we're talking about vaccines to help rapidly progressing acute invasive aspergillosis in neutropenic mice. So this is the model system for people who are profoundly immunocompromised. So you can imagine if you take away the immune system of a mouse infected with aspergillosis, it takes some kind of powerful vaccine to stop that infection with no assistance from the mouse's, or minimal assistance from the mouse's immune system. So they're really pushing it there. But nonetheless, it's still showing promise in enhancing the immune response. You're stimulating quite a poor immune response, but still giving the mouse, hopefully the patient, increased chances of recovering from such an infection. This is the one that was talked about back in 2012 by Stuart Levitz, who is a researcher in New York. This is something that's very basic. You take Saccharomyces cerevisiae, which is a mold. This is a particular one that is the same sort of species that you would use in food preparation, but I presume it's not the same strain. You heat kill it. So you just heat the thing to kill all the yeast, so it's dead. So when you inject it into a mouse in animal models, it doesn't cause an infection itself. And when you use it as a vaccine, it did indeed produce protective immunity against Aspergillus, Candida, and Coccidioides in animal models. So the potential is clearly demonstrated. Even a crude vaccine like this could protect some patient groups. And not just against aspergillosis, against a broad range of fungi, which is terrific. So the potential is clearly there in all three cases. What are the challenges for protecting these, for developing these vaccines? Okay, it's a bit difficult to treat something that is a long-term infection like CPA, because you have to provide sustained protection or therapeutic benefit over a long period of time. And as we know with COVID, vaccines don't always operate for a long period of time. But you could get over that by offering annual boosts, perhaps, something like that. It's very complex. You're dealing with an immune system that's highly complex. We've already covered the fact that CPA patients have underlying lung conditions and subtle immune dysfunctions. So you could end up with a situation where you've developed a vaccine that works for some patients, but not all patients, because you haven't got those with a particular immune dysfunction. So you have to try and make sure you're getting everybody, so that increases the complexity. And at the moment, there is no clinical trials. In fact, there have been no clinical trials specifically targeting vaccine development for CPA, which is not good. Future directions, how much time have we got? Okay. Future directions, therapeutic vaccines are being explored. We could be looking at combined therapies, which we've mentioned, where you combine a therapeutic vaccine with maybe one that's not hugely effective, like that broad spectrum vaccine that's very simple to make. Maybe you could trial those in humans and you'll get maybe a 20% response. But if you combine it with an antifungal drug, you might get a much higher response, and the patient would still be better off rather versus just the antifungal drug itself. And it's very much part of personalized medicine. We might find, because different patients have different immune function defects, that, as I said, some vaccines will only affect a small number of people. Maybe it's down to 1 or 2 people, and developing a personalized vaccine for your particular strain of aspergillosis is an aim for the future. Personalized vaccination, personalized medicine is very much an aim of the NHS going into the future. Okay, what about ABPA? We've talked all this time about CPA, but of course, more than half the people probably in this call are ABPA, and it's a more common disease. Similar situation, there are no licensed vaccines available for ABPA. Research is ongoing. And the similar caveat that most of the research is directed at invasive aspergillosis. Asper3 protein vaccine that I showed you a minute ago, and the important phrase in the findings was the lungs of vaccinated mice were free of hyphae and showed minimal inflammation, whereas non-immune mice succumbed to infection. Well, that's very much describing the problem in ABPA. Your lungs are full of hyphae and inflammation. So if the vaccine does those 2 things, then it's definitely got potential to treat someone with ABPA, which would be fantastic. I mentioned it, I touched on earlier about allergen immunotherapy. These are the biologics that were already in operation. This is another type of immunotherapy, of which vaccines are one, but this is allergen immunotherapy. We've already found the biologics to be very effective. This was brought up after I wrote this. This came up in the Facebook group. If you remember, people with protein allergy are hypersensitive to tiny amounts of protein and experience anaphylactic shock. Well, ABPA is an overreactive immune system as well. Could we treat patients with small amounts of Aspergillus antigens to induce some reaction, but as happens with peanut, you start with small and you build up and up and up, and you monitor and you monitor and you monitor, and you only build it up. Oops. You only build it as you build it up very slowly. Then the patient stops responding and generates tolerance to the Aspergillus. And if that was the case, then an ABPA patient who was immunologically tolerant would stop being so inflamed because of the fungus in their lungs. And we know from biologics that if you stop the inflammation, the patient feels 100% better. Maybe this is a valid approach, but nobody has been brave enough to try it as yet. You can imagine the prospect of giving someone who's highly allergic the substance to which they're allergic in tiny amounts needs to be done very carefully under very controlled conditions. And although they can do it for peanut, I know that took decades before people worked out how to do it safely. And it has yet to be attempted, but it has started to be suggested as an area of research which is worth following. If nothing else, it's a very cheap type of research to do. We have allergens that we could use now if we could think of a safe way to do it. Perhaps we need to learn a bit more about ABPA before we progress with that, but that's definitely an option for the future. Yeah, challenges, again, complex immune response. It's a very difficult thing to do. ABPA involves hypersensitive immune reaction. Making a vaccine development for that would be quite difficult. Again, there are lack of suitable animal models for ABPA, but there are reasonable models for CPA, but ABPA with an induced immune response. There is no such model, and until you get a model that you can work on, then you can't test for potential treatments and solutions. So I guess in ABPA, that is a stumbling block for now. Same sort of future directions of ABPA for CPA. So I mentioned earlier that we talked about one of the types of vaccine in 2012. That was 13 years ago. Why is vaccine development for aspergillosis progressing so slowly? Because there's no doubt it is. We developed a vaccine in 10 months for COVID, and we're not talking about something that's dramatically different to that. Why is it so slow? Well, primarily because of a combination. It's complex, as we've just been talking. It's quite a difficult thing to do. But probably just as important, maybe more important, there has been very limited funding for this sort of work. And there is little incentive for drug companies to do this sort of research because the numbers of people that they would sell this vaccine to are relatively low. They're doing it for acute invasive aspergillosis because you're talking about sort of 10 to 15,000 people a year in the UK and a lot more worldwide. So that's a market they can design for, but the chronic aspergillosis patient population, particularly CPA, is quite small, much smaller than that. So there needs to be some sort of, presumably some sort of public funding incentive for companies to get to progress this with any pace. It isn't it's complex. We've just said CPA is not easily cleared as a chronic infection. ABPA is an allergic reaction, not a typical infection. However, invasive aspergillosis occurs mainly in the immunocompromised, and they are the hardest group to vaccinate. So something that works for IA may well work for the other two, and that's, if you probably remember, that's the same situation with antifungal drugs. They're often developed for invasive aspergillosis first, and then they get tested for the chronic diseases second. So they arrive a couple of years later, normally, for CPA and ABPA. So a variety of reasons why vaccine, by a variety of technical reasons why vaccine development is relatively slow. Lack of funding is a big one. Aspergillosis is not classified as a global health authority or global health priority, so it receives less public research funding. So there's nothing much in it for private funders, and it doesn't get a lot of public funding either, compared to things like TB, malaria, and so on. Pharmaceutical investment, the potential profit is low, so their investment is low. For example, the WHO Fungal Priority Pathogens List 2022 finally gave Aspergillus fumigatus high priority, but it is years behind bacterial pathogens in investment. But at least it looks like investment is being stimulated now after all this time. Small market, low profitability. Clinical trials are difficult. It's hard to recruit large numbers of well-characterized CPA or ABPA patients unless you happen to be the National Aspergillosis Centre, of course. We would probably almost certainly be asked to participate in any trials with vaccines for CPA and ABPA because we have a large group of patients who have both, probably the largest in the world. So that helps. Our very existence helps the work. Trials are harder to run. Variability in diagnosis and disease severity makes trials harder to run. Who do you treat? Do you treat the really ill people? And the chances are probably yes, which makes it harder to attempt the trial. And there are ethical issues in testing novel vaccines in the most ill people. You must, first of all, not do harm, and potentially that's the group that we could do the most harm in. So that's a difficulty, but it's not something that couldn't be overcome, I don't think. And of course, aspergillosis, despite all the work we've done over the last 20 years, is still under-recognized. Globally, especially in lower income countries, the lack of priority means that less money is spent on and less recognition is achieved. And that's really what Gaffey's been doing all these years, is to try and persuade lower income countries to prioritize things like antifungal drugs and recognition of aspergillosis. And they've been doing that for probably 15 years now. A lot of progress, but there's still more to be done. Still, many healthcare systems underdiagnose or misdiagnose CP and ABP. Even in the UK, we're still a bit slow in some places. So part of my job is addressing that inequality in the UK, and other people are doing it throughout Europe and throughout the world. True burden is likely to be higher. So if the burden was higher, then there's more in it for a drug company to invest. So you're more likely to get the development of a thing like a vaccine for aspergillosis. So this is important. This is directly important to promoting the development of these vaccines. And that's just a summary of all of that. What can we do to change the pace? We need coordinated action across research, funding, policy, and patient advocacy, and that's where you guys can help. Raise awareness. This is what we've all been doing, and you guys have helped a lot. I know Gwyneth has contributed to several articles in national papers, and that all helps with this, communicating the scale and severity of CPA, ABPA, and IA. Once you've got awareness, once you've got this coded into your country's health system, then you start to think, well, what can I do about it? And that's when more investment happens. And we do that using things like patient stories. We're constantly asking you all for patient stories, and this is why. It's not just spurious what we want to talk about you. We want everyone to know how difficult life can be when you've got aspergillosis. And we spend a lot of time promoting on social media and on our websites. A push for routine surveillance and reporting in public health stats, which is what I just mentioned. Raise awareness. You can help by supporting public campaigns when they happen. These likely come from Gaffey, come from the Fungal Infection Trust, come from Aspergillosis Trust. Anyone that's actively promoting awareness nationally and internationally. So British Lung Foundation, European Lung Foundation, these are all worthwhile organizations to work with. We will help as much as we can because it's in our interests to promote the work of NAC and aspergillosis across the world. Sharing local data and patient impact in health meetings and online. Our doctors do that. Beth is about to go to a big conference in Manchester at the end of the month, and she'll be promoting the work of NAC and also awareness of aspergillosis, and talking to our doctors are going with her to talk about when to try and convince other doctors when they should start thinking about, well, actually that might be a fungal infection and not a bacterial infection. Worsening asthma might be due to aspergillus, and not give more and more steroids, that type of thing. Used to be a lot worse, it's improved a lot, but we're still working on it to this day. And writing to MPs, healthcare trusts, local media, all helps. Advocate for research funding. I guess that's something you can do through your GP. Join or support there are rare disease networks that we occasionally promote on Facebook, and that's why, and patient organizations. When people start to work together, they have a much bigger impact. Engage in patient and public involvement for research proposals. You already all, quite a few help with that, which is fantastic, because once awareness is there, once we've got the cases, we need to do research, and That's how it happens. Encourage inclusion of vaccine research in national respiratory disease strategies. So things like you will see occasionally happening NHS England or BLF, more likely BLF, British Lung Foundation, will be asking people to comment on their 5 years or 10-year strategies. And it's useful to support those. I think my battery's running out. Okay, that's mainly for scientists. Strengthen patient-led advocacy. There's been a successful approach for cystic fibrosis, HIV, and COVID. We can build public-facing platforms, lobby for clinical trials. So at this stage, I was going to actually start talking about a discussion, so we could discuss maybe a way forward for doing these things, but I'll have to wait. But we will try and organise a way for you to help with all of these things, maybe in a more coordinated way than just talking about it in a talk like this. Encourage policy change, etc., etc. We are the largest community of aspergillosis patients globally. We can work with Aspergillosis Trust and all the others, coordinate with GAFI and rare diseases. And the discussion point was, are we doing enough? A lot of people are doing quite a lot. So you do help us a very great deal.